Stem cell culture. Stem cells are undifferentiated biological cells that can differentiate into specialized cell and can divide through mitosis to produce more stem cells. They have two important characteristic features which are self-renewal and potency. Self-renewal is the ability to go through numerous cycles of cell division while maintaining the undifferentiated state. Potency is the capacity to differentiate into specialized cell types. This picture here shows the example of a stem cell. There are two kinds of stem cells, which are embryonic stem cells and the other one is adult stem cell. Embryonic stem cells are those which are derived from early embryo which is developed in vitro fertilization. The inner mass of the undifferentiated cells in the embryo are isolated and they are considered as embryonic stem cells. Adult stem cells are those which are undifferentiated and found throughout the body that divide to replenish dying cells and to regenerate damaged tissues. They are also known as somatic stem cells and they are derived from sources like bone marrow, embryos, amniotic fluid and umbilical cord. Microbial protection is one of the applications of biotechnology. The microbial products of everyday uses are vaccines, antibiotics, vitamin B12, enzymes, insulin. Vaccines. They are killed or live germ suspensions which induces the production of antibodies and bring forth immunity. Antibiotics. They are chemical substances derived from microbes like fungi, bacteria, etc. employed to kill the infectious germ and cure a disease. Vitamin B12. Biotechnologically synthesized vitamin B12 is used to cure anemia. Enzymes. Biochemically significant enzymes are derived from microbes. Insulin. Diabetes is treated by biotechnologically produced insulin. Next we are going to see is biosensor and biochips. Biosensor. It is a device consisting of immobilized layer of biological material such as enzyme, antibody, hormone, and it has a contact with the sensor. This sensor converts biological signals into an electrical signal. It is used for medicinal and industrial purposes. Example, checking blood glucose level, pollution in water, or toxic in body substances. Next is biochips. Biochips are microchips which are developed by techniques of biotechnology. In future, biological computers will be developed using biochips. Biochips will be useful in defense and medicine. Gene therapy. Gene therapy is a means to treat or even cure genetic and acquired diseases like cancer, AIDS by using normal gene to replace a defective gene. It can be used to treat defects in the somatic or gametic cells. Example, curing insulin-dependent diabetes by replacing beta cells. Types of gene therapy. There are two types of gene therapy. The first type is somatic gene therapy. In this, the whole genome of the recipient is changed, but this change is not passed along to the next generation. The second type is germline gene therapy. In this, the egg and the sperm of the parents are changed for the purpose of passing the changes to the next generation.